Welcome to Mr. Chalk's Revision Tips. In this video, we will look at osmosis, osmosis in cells, and osmosis explained. So osmosis is the term that describes the movement of water through a semi-permeable membrane from an area of high concentration to low concentration. So water moves towards a high concentration of solute with the effect of equalising the solution concentration across a permeable membrane. So from the point of view of the water, it undergoes diffusion, moving from an area of high concentration to an area of lower concentration. Osmosis is also a form of passive transport as it does not require any energy. Water potential measures the concentration of free water molecules. It is a measure of the tendency of these molecules to diffuse to another area. The more free water molecules there are, the higher the water potential will be. So water diffuses by osmosis from a region of high water potential to a region of low water potential. So osmosis can therefore be defined as diffusion, like we've already said, but the water is always going to go and move from a region of higher concentration of water to a region of lower concentration of water. So if we look at the diagram below, that shows 3 cells. Right, each of those has a certain water potential. So 10 being the highest, 5 being the lowest. So if we have a question that says in which direction do you think the water will move, the water is always going to go and move from a region of higher concentration to a region of lower concentration. So when a cell is submerged in water, the water molecules pass through the cell membrane from an area of low solute concentration to an area of high solute concentration. So if a plant cell is placed in a solution of water with a lower water potential, water will diffuse out of it. So this causes the cytoplasm to shrink and the cell will become flaccid. If enough water leaves the cells, the cytoplasm will pull away from the cell wall, so the cell will become plasmidized. So, since the cell contains various biological molecules, such as sugars and salts, they have a water potential lower than 0 kilopascals. Right? A kilopascal is just the unit that we're measuring water potential in. So water may move into or out of the cell depending on the water potential gradient between the inside and the outside environment. So when water diffuses into the plant cell, when it's placed in a solution of higher water potential than inside of it, the cell contents will expand. However, since plant cells are surrounded by a strong cell wall, they will not burst, so the cell contents will push against the cell wall and the cell will become turgid. So if we look at this question, so the diagrams show the same cell placed in different solutions. So diagram A shows the cell in a hypertonic solution, and diagram B is the same cell in a hypertonic solution. So what is a hypertonic solution? So it's one where it's more concentrated than the cell or the cytoplasm. So explain what happened to the plant cells in diagram B. So water has moved out or has left the cell by osmosis from a dilute concentration to a more concentrated concentration of the solute via the semi-permeable membrane. So therefore the cell will shrink away from the cell wall. Animal cells will also change if placed in different solutions. So some red blood cells are put into a hypertonic solution. So describe what would happen to these red blood cells and explain why this is different from what's happening to the plant cells in diagram A. So things like water would enter the cell by osmosis, but they would expand and burst or lyse. So water leaves the cell and it would shrink if it was a hypertonic solution. So animal cells have no cell walls, so therefore they have nothing to hold them in place, which is why they would burst. So students put plant cells into two different strength solutions, A and B. So if we look at the diagram and describe two ways in which the cell in solution B is different from A, it's simply things like the vacuole is smaller, or the cytoplasm is shrunk, or the gap between the cytoplasm and the cell wall is increased, or the cell curves inward. So if we look at a osmosis investigation, right, we can go and set something up like this. So the following experiment investigates the effect of different concentrations of sucrose on a potato tissue. Right, it could also be carried out using a sodium chloride solution or various other solutions. So when we go and we carry out an experiment like this and then observe the resulting change, we can look at the change in mass. So we can see there that as the sucrose solution increases, more water would go and move out of the potato cells, causing their mass to decrease. But in a relatively dilute solution of sucrose, so either 0 or 0.1, there'll be a higher water potential in that solution, so therefore water will move into the potato cells, and that would cause an increase in their length. So another experiment could be something like this. So a student carried out an investigation using chicken eggs. So they placed 5 eggs into acid for 24 hours to dissolve the eggshell. They measured and recorded the mass of each egg. So they placed each egg into a separate beaker containing 200 centimetres cubed of distilled water. After 20 minutes, they removed the eggs from the beakers and dried them gently, and then re-weighed their mass. So the results are shown in the table to the right. So another student suggested that the results for egg 4 was anomalous. Do you agree with this? So we go and we look at those results. We would agree to it because it's much lower than the others, right? So we can discount that because it doesn't follow the pattern. So explain why the masses of the eggs have increased. You'd need to talk about water's entered by osmosis from a dilute solution in the beaker to a more concentrated solution in the egg through that partially permeable membrane. And explain how the student could modify the investigation to determine concentration of the solution inside of the egg. You need to be saying something like use 5 or more different concentrations of salt solutions, then plot them on a graph, and then you could determine where the concentration was the same, because it would be at the point where your line passed through the axis and would show, or pass through the axis at a 0% change.
Thanks for watching.